Let's come to the first characteristic of enzymes. Almost all enzymes are proteins. Enzymes are mostly proteins, so they are formed out of 20 different amino acids like other proteins. So there are 30,000 proteins in human. Out of that, 50% exhibit enzymatic activity. So about 15,000 different types of enzymes are found in human. These proteins are the most diverse group of molecules, and so enzymes are most diverse in their structure and function. However, during the evolutionary process, RNA was the first molecule to be evolved, and RNA was the first molecule to catalyze chemical reactions. After this evolution, when this cellular evolution progressed, DNA and proteins were synthesized, and DNA took over the function of genetic material, and proteins took over the function of catalysis. However, there are some RNA molecules which keep on catalyzing chemical reactions. So these catalytic RNA molecules are called ribozymes. So ribozymes are catalytic RNAs. So one of the one example of ribozyme is peptidyl transferase. So this enzyme forms the peptide bond between two amino acids during protein synthesis. Since enzymes are proteins, they follow all the principles what proteins follow. So if we can understand the structure and function of proteins, we can understand the structure and function of enzymes. So proteins, when synthesized, they form the primary structure. It is a linear chain of amino acids. So they form the secondary structure gradually. The secondary structure of protein is formed by local folding of the protein chain. So the secondary structures are like alpha helix and beta sheets. So there are other secondary structures, so I am not going to that. So this secondary structure again interact with each other to give the tertiary structure. So if the protein is having a single polypeptide chain, it will form a tertiary structure. But if it is formed out of multiple polypeptide chain, in other words, if it is a multi-subunit enzyme, it can have several subunits joined together. So they can form quaternary structures. So enzymes have one active site to which the substrate binds. It can have other sites to which different regulatory molecules bind, but all enzymes must have one active site. And this active site is formed by about 5 to 10% of the total protein, whereas the rest of the protein play a structural role to preserve the three-dimensional structure of the active site, which is essential for the activity. In the next video, we will go to the second characteristic of enzymes.